Hello, everyone, and welcome to today's live broadcast, Cryo Electron Microscopy, Revolutionizing the World of Structural Biology and Healthcare, presented by Marc Storms, Product Marketing Manager, Life Sciences Business Unit, Materials and Structural Analysis, Analytical Instruments Group, Thermo Fisher Scientific. I'm Alexis Krauss of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know that you're having a problem. Please join me in welcoming Marc Storms. I will now turn the presentation over to him.
Thank you very much, Alexis, for introducing me and giving me the opportunity to introduce you into the world of cryo electron microscopy. In this presentation, I will cover actually a number of topics. First, I will quickly introduce into Thermo Fisher, the role of Thermo Fisher in electron microscopy. And then I will switch to explaining a little bit what currently drives structural biology, the complementary technologies that bring their individual values to the structural biology questions, scientific questions that people actually face today. I will spend some special attention on the latest, let's say, last couple of years revolution that Cryo-EM has undergone through and the recognition that technology basically has recognized, has actually obtained. I will show some unique applications data where Cryo-EM has made a difference. I will also talk a little bit about the unique single particle workflow concept that Thermo Fisher has developed. And also the challenges that come along with this workflow. And if you are interested after hearing my story for, let's say, 75%, I'll cover also some slides on how you can engage into Cryo-EM, what is the best way to get in contact and adopt the technology, basically. And as already introduced, there is also summary and conclusions and Q&A session at the end of the presentation. First of all, a quick introduction into, let's say, Thermo Fisher, the groups, and where actually electron microscopy belongs to. In principle, is actually electron microscopy part of the Analytical Instruments Group, which also includes equipment like spectroscopy, mass spec, and chromatography equipment. This Analytical Instruments Group is roughly representing 22% of the total revenue of 21 billion within Thermo Fisher. And with that is actually also the area that is focused mainly on, let's say, the advanced equipment for analysis and structural biology analysis. The basic question, of course, is why is actually structural biology important, and where do we see actually a role within structural biology in the upcoming years? And the main rationale behind this statement is the fact that people grow older. Luckily, people grow older, obviously, but when people grow older, that also comes with visible and invisible chronic diseases, elderly diseases like Alzheimer's, atherosclerosis, and diabetes. And people, of course, ideally would like to live long and happy without too many of those diseases. So to give an indication, currently around 40 million annual deaths actually are occurring from diseases which are related to actually elderly specific diseases. In order to actually avoid those diseases from occurring, and we need to understand the fundamentals of the disease mechanisms much better. And if you say this, it also automatically means that we have to understand critical metabolic pathways. And it all comes down to the biochemistry or molecular biology level of understanding the building blocks of life, understanding how a protein behaves as an individual protein, but also as a protein which interacts as a complex with other proteins, or with DNA, or with membranes, or in the context of a cell. And if this is understood, then it can also play a role in the development of new drugs and more efficient drugs in general. And that's basically the driver between current research in structural biology. If we talk about structural biology, there are actually a lot of technologies that play a role here, or actually form part of the structural biology technology base, so to speak. The Cryo-EM is one of them, and I will talk in detail about maybe the most important application right now, which is called single particle cryo-TEM. But also cryo electron tomography will emerge as an equally important technology within now and a couple of years' time. But also NMR, very classical technology, and X-ray crystallography are very, very important technologies next to X-ray microscopy and of course light microscopy. Basically, they have their own scale in the landscape of technologies, and as Roger Kornberg rightfully said, each technology brings something important to the table, and the combination of the technologies makes the sum actually most powerful. So it's all about complementary technology. It's not that Cryo-EM necessarily will replace some of these technologies. Everything adds a piece of the puzzle in this respect. What is this piece of the puzzle? In some respect, you can say the same, of course, about the workflow from the basics of life, the genome, towards the full human body, let's say the large volume analysis or the cell or tissue biology. Everything starts, of course, with the genes, the genomics, the translation of the Human Genome Project, for example, into various proteins that are expressed by these genomes. Currently, we're in the status of exploring the shape and the function of these individual proteins and how they respond if they interact with each other in a cell, for example, in the specific disease case. And this is where Cryo-EM single particle analysis kicks in. It is obviously very important that the information that is revealed by performing crystallography and performing Cryo-EM single particle analysis is positioned, translated into the cellular context. Obviously, it's interesting to look at one individual protein complex, but it's only functional when you look at it in the context of the cell. And that is actually where Cryo-EM tomography will kick in. The transposition of high molecular structure biology information obtained by SPA and crystallography into the cellular context. And ultimately, of course, that is also the same story is true, fits into a cell, into a tissue, into an organ, and into the total body. For now, actually, in this presentation, again, I will focus on what I mentioned already, single particle analysis. That the technology actually has become very well recognized the last couple of years, has already become clear if most people have read literature. In 2015, single particle analysis was actually awarded as Method of the Year, mainly because of what is called the resolution revolution, the sudden increase in obtained resolution of proteins and protein complexes due to some very differentiating technologies, which I'll get back to later on. And this year, actually the end of last year, I should say, the Nobel Prize for Chemistry was awarded for the pioneers in cryo electron microscopy, being Jacques Debouchet, who mainly focused on sample preparation, and I will talk about it a little bit more in the next couple of slides. Richard Henderson, who was the first to really acquire and make a 3D image of a protein at atomic resolution, and did a lot of development on detectors as well. And Joachim Frank, who's basically playing a role at the backside of the single particle workflow, the image processing part. He was the one who developed, among things, algorithms that make it possible for fuzzy images, cryo images, to turn into a 3D reconstruction. So the technology is really, you can say, well recognized, and there are a few technological reasons basically for this revolution, if you like to call it that way. And a couple of these technologies have been invented or co-enabled also by Thermo Fisher and FEI. It's basically predecessor. The most important one to mention are the direct electron detectors which have an extremely high sensitivity, which have actually the capability to look at very low electron dose image information, which makes it actually possible to look at very sensitive material at the maximum possible resolutions. But also the platform and the acquisition and the automation is very important. As I will show you in the next couple of slides when I explain the principle of single particle analysis, it is a highly automated process. Less and less manual interference is needed actually in this case, and development of specific software also plays a crucial role in here. And last but not least, of course, it's all about the reconstruction software. A lot of academic institutions have developed various software packages for reconstruction of single particle acquired data sets and turn them into 3D reconstruction. I think the subset of all these technology breakthroughs have made it possible to also create a scientific impact actually on the community. And I also will give a few examples of that. A structure to function analysis, a better understanding of actually disease mechanisms, but also even target drug discovery is now in the exploratory phase, thanks to basically the technology breakthroughs as I just introduced. And obviously, as already said, there are complementary technologies like XRD, NMR. They have actually a long history, a long tradition already, played a crucial role. There are a lot also of data in the protein database obtained by using XRD and NMR, but there is actually an interesting sweet spot where Cryo-EM fits in. If you would currently, let's say, want to reveal where Cryo-EM stands in terms of the achievements, then you can basically, if you want to simplify the entire story, say the best possible resolution that has currently been obtained by doing Cryo-EM single particle analysis is already sub 2 angstroms, 1.8, it's actually slightly better already, 1.5 into that direction. Scientists already have gone with specific samples. You can see individual water molecules, for example, and aromatic rings and side chains already at this resolution, extremely good. But also the capability of Cryo-EM to look to large membrane protein complexes, and I talk really extreme large membrane protein complexes like this example on this slide, the respirosome complex of 1.7 megadaltons, which consists of 81 proteins in a complex. That's really unique for Cryo-EM. Cryo X-ray crystallography is in this respect not capable of crystallizing that large portions of proteins in a complex manner. And this is also where actually Cryo-EM makes a difference. But also very small proteins can be analyzed. To a couple of years ago, 5-10 years ago, Cryo-EM was valuable if you would look to steady state structures, large structures like viruses, megadalton complexes, but there's a lot of course of science into the smaller protein complexes, into smaller proteins in general, sub 250 kilodaltons and even sub 100 kilodalton proteins. And thanks to another technology breakthrough, the phase plates, it has also become possible for small proteins to be visualized, and this example is of a 64 kilodalton hemoglobin protein. So to summarize it, resolution gets better, proteins get larger in terms of the complexity that can be revealed, but also on the other hand, proteins can get extremely small and still being able to be imaged at high resolutions. Now if you would look to, and that's more or less confirming my previous message, to the protein database, for example, then a lot of structures, probably more than 90% of all the structures in there, 85% has been obtained by X-ray crystallography and nuclear magnetic resonance, the two other techniques that I introduced already in my presentation previously. But those technologies have one, let's say, limitation. They look to the building blocks of life. They can analyze the building blocks of a protein, the monomer or dimeric structures. Whereas the real scientific interest is in looking to large protein complexes, because they gain new biological insights. Understanding how a protein complex works is understanding, let's say, real life in the context of a cell, and it also means new biological insights. And this is the sweet spot where Cryo-EM nicely fits in, where a lot of publications that you currently see in reality is focused on. So just to give you 3 examples of cases where proteins where actually Cryo-EM has played a crucial role recently, and there are many more examples, but I just picked 3 of them randomly. A study of, let's say, dynamics. By doing Cryo-EM, you can capture various isolated states of a protein and turn them into information that reveals something on dynamics. On the other hand, I already mentioned that complexes, that's really what the value is of Cryo-EM. You can look to large protein complexes. And also short time to result is an important argument. In some cases, Cryo-EM can really be much faster than the alternative technologies, and I'll also give you an example on that one. The first example is an example from the publication in Science from, let's say 1 year ago roughly, and it's about a protein, ATM protein, that plays a role in, let's say, repairing DNA that is damaged by for all kinds of reasons. Luckily, we have this DNA repair mechanism, otherwise you could trigger all kinds of mutations and maybe even cancer. Now, it's important to understand how this ATM protein, this DNA repair protein, works. And we know from analysis by Cryo-EM that there is a kind of switch, a molecular switch, which switches the protein on and off into an active and into an inactive state. Well, understanding this molecular switch is of course very important, because again it reveals something on the fundamentals of life, and it can also create insight into dynamics that could contribute to the production of more efficient drugs against cancer, for example. So that's actually the rationale behind this research, and thanks to EM, basically the next level of information could be revealed. Similar story, but on a different league, is true for looking to this the tau protein filament aggregation mechanism of which plays a role during Alzheimer's disease. Like a lot of similar type of diseases, Alzheimer's is also characterized by creating large protein stacks, protein filaments basically, which can cause kind of plaques in the brain in this case. And by analyzing material from a deceased patient, Alzheimer's patient, Cryo-EM could do something unique, and this is the work of Anthony Fitzpatrick, quite recent work last year published in Nature. We could distinguish for the first time two different types of filaments that actually are playing a role in the aggregation process and aggregation mechanism of this plaque formation. So understanding what the building blocks are, what the different monomeric states are, as if you like, of the filaments, you can also actually get a better understanding maybe in how these plaques are formed and how this aggregation process is formed. And understanding how aggregates are formed in the brain could maybe lead to a better understanding on how to design drugs, or maybe even a way to prevent these aggregates from forming. These are only tiny steps, but important steps, and the basic block, you can say, has been revealed by performing electron microscopy in this case. Another example is, that's the last one for now that I will share, is the Zika outbreak. Everybody, of course, is aware of what happened in 2015, 2016. Terrible disease with a lot of serious consequences for embryonic and young babies and children. The best way, of course, is to make sure that you develop a vaccine against Zika, and then hopefully the problem can be solved. And for developing a vaccine, again, it's also important that you understand how the virus structure looks like from the outside. So again, a better understanding of various monomer and dimeric structures, in this case dimeric structures of the virus. While the individual E and M structures were known by crystallography, that was already revealed, but the overall complex organization was not known yet, and by performing Cryo-EM, that could also be revealed. And that has actually led to, in 2016, to revealing the first virus publication structure already at 3.8 angstrom resolution. That was already quite decent to start evaluating and experimenting with the first vaccines. And that happened at the end of the year, first vaccine testing, and the phase 2 trial testing is already, since March of last year, already in progress. So thanks to Cryo-EM, you can say that the process of developing a vaccine has been accelerated, and hopefully when the next outbreak of Zika is, there is actually a final solution for it. And similarly, of course, there are more stories along the same lines of what I mentioned here, maybe with respect to Ebola, maybe with respect to HIV. So Cryo-EM really, actually from an application point of view, is showing that it is capable of moving the needle, of shifting the boundaries of knowledge. Why is Cryo-EM so important from, let's say, an experimental point of view? Why is it, let's say, interesting compared to the alternative technologies, which definitely also still have a lot of value and are still used a lot? One important example is when you do Cryo-EM, the word already says it, you're vitrifying, you're cryo-fixing actually the samples. And cryofixation is extremely important because it gives you the best possible structural preservation. If you do this in another way, by using chemical fixatives, there is a high chance of creating all kinds of artifacts. And you want to really look to reality in the most accurate state as possible. And this is where vitrification plays a role in. Secondly, which is also an example, if you do Cryo-EM, there is no need to first create a crystal or do an isotopic labeling like you need to do for NMR. In principle, it's just a matter of getting the protein purified into the proper concentration and then vitrified by using liquid ethane and propane. So it's a little bit easier on paper at least, because you miss part of the complex crystallization and labeling process. And in general, you need only very little, last advantage, little aliquots of material. So a few microliters, actually even nanoliters of suspension are in principle already enough to perform Cryo-EM. That makes it actually very, very attractive. And the next actually part of the presentation, I have a short video by a research fellow in Sweden who recently adopted Cryo-EM. And I'd like to ask my moderator to start the video and see what he has to say here.
The overall scope of our work covers processes related to gene expression in organelles, such as mitochondria and plastids. This includes fundamental questions such as how proteins are synthesized, folded, and assembled into macromolecule functional complexes. It also includes investigation of evolutionary forces driving the development of gene expression. And another fold of our work is the investigation of the medical aspect of how gene expression can be therapeutically targeted. And finally, we also work on bioenergetics-related questions such as sunlight-driven light conversion, which is also linked to gene expression in chloroplasts. We address these questions with structural biology, which allows direct visualization of cellular mechanisms and therefore provides the most efficient way to reveal molecular bases. Specifically, we employ electron cryo-microscopy, where biological samples are flash frozen fast enough to preserve their native state, which allows investigation of their structure in the context of the cellular function. And we are very privileged here at the Cryo-EM lab to have access to the most advanced technology that allows visualization of molecules up to atomic resolution. But beyond the high resolution, perhaps the greatest advantage of Cryo-EM is the identification of multiple proteins, as well as multiple protein conformation from a single specimen, so that it really allows to record a movie of molecules as they perform the cellular function. This can be then extrapolated to medical applications, where recording structures and reactions in the presence of therapeutic compounds. Now, the world of molecules is beautiful to look at, but it's even more beautiful to be able to understand it. And what is particularly challenging in our work is to interpret the molecular structures into biologically meaningful information. This work is also the most fascinating, as it's really like solving a jigsaw puzzle where we are trying to assemble structural elements together into one single image. And during this process, you have the feeling that you are the first person to reveal these molecular structures. And there is no more beautiful and rewarding feeling for structural biologists than to see how molecules are sculptured by fundamental laws of biochemistry. And this information is universal because in biology this is as definitive as it can be. What impels me to do research and to lead a research group, apart from the curiosity of course, is to watch how people developing and maturing into scientists, how they're advancing professionally and personally through their work, and this aspect I find one of the most rewarding in leading a research group. And I consider myself very privileged to be part of this collective effort and the great atmosphere that we have in the lab.
So that's actually, I think, a clear story on one of the customers that actually has adopted Cryo-EM for very obvious reasons. Now, the next part of the story, I will talk a little bit about Cryo-EM and its workflow, the specific technological steps, if you like, and what are the challenges in order to get, let's say, these challenges overcome. The first slide actually shows the process of single particle analysis, and what actually you will see in the movie in the next minute again is the process from the biochemistry part, putting a sample onto a grid, which is actually a sample carrier that you put into the microscope. Once you go into the microscope, after having vitrified the samples, you acquire a number of noisy 2D projection images, and you try from those images to select those particles of which have various orientations, average those orientations, and by averaging and basically summarizing them, if you like, you get a refined 3D model. And again, I would like to ask the moderator to start the video on the workflow for single particle analysis. So that was actually a nice animation on single particle. So what happens if you would digest actually the various, or put the various sections into perspective, and you start with a biochemical component, where you purify the sample, as I said, then you try to vitrify the suspension that you would like to investigate by using the Vitrobot. Then quite often there is a step, and I will talk a little bit more into that, of optimization needed, where you look to the protein distribution, but also the thickness of the ice instead of the proper quality to really do the analysis and the data acquisition in the end. So the process, although it looks actually pretty nice, and it is actually a very nice process, there are still actually very critical parts of the workflow which need to be taken into account. And I will quickly, in the next couple of slides, tackle those individual parts. The first challenge is get the best sample quality. After that, it's important to minimize the time on sample preparation and sample optimization, because you want to go as fast as possible to the microscope where you do the final analysis, preferably at the resolution that is needed to answer your scientific question. And that can be anything, let's say in general, something below 3 angstrom is sufficient to answer most scientific questions. On the first part of get the best sample quality, there are actually a few crucial components that are important and should be taken into account. On the first part, the biochemistry part, it's obviously important that the protein is expressed to sufficient levels in order to purify it. Once you have these sufficient levels, you need to purify the protein, get it clean enough, make sure that you get it to sufficient concentration levels that is also stable, that the proteins do not fall apart, for example. And ideally, if you isolate various fractions or various states of a protein, you would need also to capture all these states within this purified fraction. That's of course a job on its own. But suppose you get to the right stage, to the right concentration, then the next phase is to do the vitrification. And the vitrification is something that has been automated. Let's say in the last 10-15 years, various vitrification devices have been developed. One of them is the Vitrobot, and there are a few more which kind of remove the critical parameters that determine the variations of the right ice quality. You would like to get repetitive ice quality every time you do the vitrification step. And it means repetitive in this case, that the ice quality that you obtain when you plunge freeze your sample into liquid ethane or liquid propane, that needs to be at the right constant thickness. But also the ice quality is very important. It needs to be icosahedral ice. It needs to be the best ice quality, amorphous ice. It should not be hexagonal or cubical ice. And last but not least, it should be possible also to look to the various particles in the proper orientations, because in the end you need to get all these orientations nicely classified and averaged. So there are still quite some challenges, despite the fact that to some extent the process has been automated, at least the vitrification part. There are still a lot of challenges to get from the optimal biochemistry into the frozen hydrated sample into the microscope. But suppose you get there, and of course this is something that is already pretty routinely right now, but still it could be better in the end, you would like to have a look to the quality of the sample into the microscope. You would like to see what the particle aggregation is, for example, what the concentration of the particle is. Well, this is what we call screening. And currently screening is a crucial part of the single particle workflow. How you do this, there are various ways to do it. Typically, one of the steps that people apply a lot is by using a piece of the sample, apply a heavy metal stain to it, which we call negative staining, and by looking to the heavy metal contrast in the microscope, typically a smaller microscope, a Talos L120 for example, or another 120 kV microscope, gives you the information about the protein aggregation or the particle distribution. If that turns out to be good, then another screening can be done under cryo conditions, where you also look at the same parameters, but in addition to that, you also look to the quality of the ice. Again, it needs to be amorphous, but also to the thickness of the ice. This screening process is a trial and error process in general. You can be lucky by getting directly to the optimal stage and then directly go to the data acquisition part, which you then can do on either a glacius microscope, which I'll shortly introduce afterwards, or a Titan Krios, but it can also take a little bit longer. Depending on how good your biochemistry is, it takes faster or longer in order to get to the real acquisition state. So important is, the more efficient your screening is and the more efficient your sample preparation is, the shorter the time to sample optimization is. Second phase, and this is where the conductivity plays an important role, once you have done a proper screening on one of the microscopes, ideally And sometimes, not always, but sometimes you would like to use the same sample or batch of samples and put them onto the microscope where you want to go, for example, for high resolution. But that is important, that the same sample can be connected to the microscope without any contamination that you can capture in the air. And this is what we call contamination-free sample transfer. So there are two important key steps in the first phase of the workflow. It's the sample preparation quality, and it's the connectivity between the screening device and the open microscope device. Now for the screening job, we have introduced a microscope last year, which is called the Glacios. The Glacios, the word already says it, has something to do with glaciers, with ice, and that's also what it does. First of all, it's designed to analyze the quality of the ice, as I mentioned before, ice thickness and the quality of the ice. But it also actually allows you to obtain reasonably good resolutions. Current record that we have is already 2.5, 2.6 angstrom, and we are improving on that. So this is a 200 kilovolt microscope, FAC tip for those who are actually experts on microscopy. It has an autoloader to load samples, to load multiple samples actually at the same time in the microscope, and also efficiently screen those samples and select the proper sample for further analysis. Another advantage of this microscope is that the Glacios is pretty small. It's not a very high microscope, so it also fits relatively easy into many lab rooms. Now this is just one result, one of the first results we obtained on the Glacios on apoferritin, a standard sample that we use here. You see on the left side a typical noisy cryo image of particles which looks nicely distributed in general at a good concentration. Second picture shows the classified, so the average particles of the same orientation, which are basically add up and multiplied. So you see much more information, the noise is slowly disappearing. And on the right side you see the colorful 3D reconstruction of the protein. In this case it's 3.3 angstrom, but we are already now at 2.5 angstrom by using the same instruments. So in principle with the Glacios, besides screening, you can get to resolutions of 2.5, 2.6 angstrom in the 2.5 angstrom range within a few days. You see the times indications below in this slide. So as I mentioned, this connectivity is really something which is very important, and also the microscopes that Thermo Fisher builds and designs are made in such a way that the connectivity is also optimized. So we have a special grid in the sample preparation part on the Vitrobot. You see that we have a grid, that's a sample carrier, which is put into a ring, you can say. That is what we call the autogrid ring. This autogrid ring is loaded into a cassette, and the cassette actually can be transported in what we call a capsule covered in liquid nitrogen to one microscope, the Glacios. So the autoloader and the autoloader components, which is the grid, they basically ensure the optimal mechanical connectivity from the screening microscope to, if needed, the high resolution microscope. So in case you get to a good sample and you would like to push the resolution beyond the 2.5 angstrom level, let's say the direction of 2 angstrom for example, in that case you need to go to a centralized service facility or to your own Titan Krios instrument, and then you can simply take the nanocap with the samples and put them onto the microscope. So there's one connectivity component, mechanical connectivity component, which allows you to do free, contamination-free connection of the samples. Second connectivity component is the software. There is also a single particle acquisition software which allows you to acquire data, to facilitate the screening process, and to select those areas of a grid which are useful to do further processing. And all the microscopes that Thermo Fisher designs have the same software. So we ensure by having this philosophy both for a hardware and for a software connectivity optimization step, by having the software and by having the autoloader ready to optimize the workflow process here. Talking about this high resolution microscope, we call this the Krios. The Krios G3i is our latest model, and the Krios G3i actually is the next evolutionary phase in the autoloader-based Cryo-EM microscopy. It allows you to load 12 samples at the same time thanks to the autoloader. We can do batch screening here. We can easily, let's say, use the instruments. There's a lot of automation software into the system right now. But also Optically wise, this microscope is capable of easily achieving resolutions better than 3 angstroms, and routinely, as was shown in the next slide, 2.5, 2 angstroms, and even better beyond that. That's not unrealistic depending on how the sample actually has been isolated, and the type of sample, the heterogeneity of the sample, plays also an important role here. And again, an example for also apoferritin, that's a standard actually use case example, but this case taken on a Titan Krios. This is done with a Falcon 3 electron detector. I mentioned the electron detectors in the beginning of my presentation. This is actually the detector that Thermo Fisher is developing, the Falcon 3 electron counting type, together with the Falcon, with the Krios G3i actually. Gets a 2.2 angstrom, but as I mentioned in the beginning of my presentation, routinely 2.5 angstrom can be obtained, depending on the sample, of course. But the world record is around 1.5-ish, 1.8 angstroms already. So optically, the Krios goes actually one step beyond the performance, at least right now, of the 200 kilovolt FEC systems in general. Well, if you now, after hearing this story, have become interested in Cryo-EM as technology, and you wonder how can I engage, how can I adopt to this new type of experiment, a new type of application, then there are various ways to do it. You can team up with an academic partner that happens to do already Cryo-EM for some time. You can happen to go to a service facility, centralized service facility. There are more and more synchrotrons currently that have adopted and purchased microscopes in order to obtain data that can be used for reconstruction of data. But you can of course also buy your own tool set. And basically there are two different configuration flavors that you can obtain. You would advisably need a vitrification device like a Vitrobot to do the essential vitrification and fixation of the sample. The glaciers for sample optimization screening would be advisable, and if budgetary wise it is possible and you would definitely need it also to push that to the highest possible resolutions, a Titan Krios would be a nice package. Then you ensure the total connectivity of the tool suites of instruments that do the vitrification, the screening, and the acquisition itself. If you would actually require the expertise in order to get acquainted to Cryo-EM, Thermo Fisher has taken an initiative to develop what we call accelerate packages. There are various flavors of accelerate packages, and I will talk a little bit about it in the last part of my presentation within the next 3 slides. But that basically allows you to make a quick start into a new technology and to get to high productivity ready in a pretty short time frame. That's actually one approach that you can go. That's basically the full suite approach. There's actually maybe even a more attractive approach to start with, and that is actually that you focus on investing into a Vitrobot and the glaciers. That's probably much more attractive from a pricing point of view. The glaciers, as I mentioned, is capable of screening very efficiently, so you can find your optimal sample, but also the glaciers is capable of getting to the desired resolutions if your resolution is in a 2.5 angstrom range and not better than that. So depending on your scientific case, that could be good enough. And then, if needed, you could go to a centralized service facility. If you would happen to go to a Titan Krios, that would then be the next phase. But in-house, you could basically start your own glaciers Vitrobot combination. So that's another approach that you could use in adopting the technology. Basically, it's actually the philosophy of Thermo Fisher to have as many glaciers and Arcticas, Talos, the same instrument, in 2 facilities next to a Krios. The Krios is of course an instrument that is run quite often 24/7. You want to use it to the maximum, in a maximum optimal state. That means you want to use it for acquiring high resolution images. And a glaciers is a kind of feeder tool into the Titan Krios. So you would like to have a glaciers to optimize your sample, isolate your sample, and then go to the Titan Krios for optimization. And this slide shows a lot of centralized facilities or Cryo-EM facilities in general at universities, institutes to Europe, Asia, and the US, where you have those instruments where you get connected to. So the approach of linking yourself to an academic partner is very well suitable. There's a high footprint actually of instruments currently in the field. The last part I would like to talk about, I mentioned it already on one of my previous slides, is actually the accelerate packages. So as more people want to engage into cryo microscopy, it's also important to arrange for sufficient support to make sure that people can get trained in an official and an optimal way. So we basically have defined what we call a customer success management strategy, which contains of a number of components besides a site survey portfolio and an IT support portfolio that helps you to analyze your laboratory space where you would like to position your microscope. We also have taken the initiative to start together with a few academic partners, Cryo-EM schools, which actually are very intense schools, 4-week practical and theoretical training courses held in the United States and Europe, and will also be held in Asia in the future, supported by an online training guidance, which gives you in the end a certified exam or proof that you actually are capable of running an electron microscope. The entire workflow is covered at those Cryo-EM schools, from sample preparation all the way up to data reconstruction. So that's actually what you can do in order to get educated, and the advice we can give basically to start with a Cryo-EM experiment and adventure. Besides that, we've also started what I already mentioned, that the Cryo-EM Accelerate Portfolio Package Suite, and this contains 6 different and new features that can help you to get more acquainted with the technology. It contains of what we call a workflow validation test. This is basically a test that we perform together with our experts in the field to show on a real biological sample on your microscope that high resolution data can be obtained, and we use apoferritin for that as an example. I showed that sample already 2 times before in my presentation. But there's also a set of hours that you can ask for on-site application support. People, experts from the nanoports in Eindhoven or the United States, they can visit you and train you for a couple of months or weeks, depending on how much training you need. This is all flexible and can be determined and tuned into your personal needs. There is obviously remote application support, where you can get for offline support, talking directly to one of our experts or to service. One of the real new things that we've done is the workflow app. The workflow app is actually a kind of best known methods guide. It's a very nice interactive app which you can run on your iPad and basically on your tablet, and you can look through all the individual processes, starting from vitrification all the way up to the processing part. If you have specific questions, you can really look those questions up. You can ask those questions into the app, and we try to actually mature the learning experience that you can get from the workflow app based on your own feedback. So it's really interactive to that extent, and very useful. Our users find. One of the new things that we have done is what we call the D2I, or the system monitoring. All the microscopes in the field can be hooked up to, let's say, a system which can pre-actively monitor the performance of all the microscopes, and service can also see which microscopes are performing properly, which microscopes need some repair activity, which microscopes need some preventive service activity. That is all possible thanks to this remote system monitoring. So the advantage is that you can minimize the risk of sudden downtime of the system, because we can do kind of preventive maintenance thanks to looking into the system that you're using. And last but not least, there's dedicated quarterly review service between you and our service organization or our customer success manager, which tells you exactly the statistics on the use of your microscope and also where things could potentially be improved. So basically, we take you by the hand. That's actually the message here, by introducing this customer success management strategy and the Accelerate portfolio rolling out to make you more successful, and especially for new users. I think that's a pretty useful exercise to do. So that brings me to my last slides in summary. If you can summarize actually where Thermo Fisher as an organization actually it is actually currently at in terms of Cryo-EM. I think we can say that the Cryo-EM in general is a proven technology. It's also a recognized technology. I mentioned that, Nobel Prize, Method of the Year. But also if you look to the output that most Thermo Fisher microscopes generate, there's a large portion of the output obtained by the Titan Krios and its predecessors like the Polara and also the Arctica. More than 1,000 structures are currently deposited, and I'm talking about 2017 data here, based on Thermo Fisher microscopy data, and increasing. the number of cryo-EM data and the protein database is significantly increasing, and probably from an absolute number it has the fastest growing rate and growing pace here. And there are a significant amount of publications, and also here it is also increasing based on cryo-EM data. There is workflow connectivity that has an advantage. We have a huge install base in the fields, a lot of Krios microscopes, Talos Arctica microscopes, and also glacial systems which are introduced are getting installed into the field. That means that we have an optimal connectivity and density to do the screening and go to any other microscope in the field for your optimal resolution performance, for example. The connectivity, I mentioned that, is ensured by, on one hand, the software, which allows you to do the screening in an optimal way, acquire the optimal data sets, and load parameters and coordinates from one microscope into another. That's what the software is capable of. And on the mechanical side, we have the auto loader and the auto grids, which allows you also to switch from one microscope into another. Of course, introducing such a microscope and having such a large database and also introducing the accelerate packages requires a lot of support troops into the field, and that's also what we currently have. We have more than 165 cryo-EM service and application specialists that take care of the maintenance, preventive maintenance, and also of the application support, and also the cryo-EM skills, obviously. And last but not least, there are multiple ways to start cryo-EM. If you're interested, I just give you a few examples. The full suite option, let's say the startups option, the connectivity with a scientific colleague or with a centralized search facility. Everything is possible these days in order to get engaged with the technology. I would like to also refer to our website in this particular request. Most information that I told you about is also on our website. And if you have more information after visiting our website, then please contact me or one of my colleagues from the Sales or Business Development Club. With that, I would like to thank you for your attention, and I'm happy to answer questions if there are any questions. Thank you.
Thank you, Marc, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window, type your question into the box that appears on your screen, and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, do you expect that Cryo-EM will replace X-ray crystallography in NMR?
Yeah, that's a good question. I don't expect that this will be the case. As I mentioned in the beginning of my presentation, every technology has a specific place, a specific niche in the spectrum of structural biology techniques. Cryo-EM is very useful where it comes down to looking to protein complexes, large molecular assemblies, whereas XRD is very good and dedicated to looking to the building blocks of life, and NMR probably into the flexible domain. So, yeah, no, it will not replace it. Cryo-EM will, I think, be a nice complementary addition to the existing technologies.
How can the Accelerate support packages be ordered?
The Accelerate support packages can be ordered actually via the regular sales process. So if you contact your local account manager, then they can help you. It's actually part of the configuration package that I explained, this full suite and the let's say startup suite package, to have it included. And depending on your expertise level, I would recommend to add this. We have 3 different flavors of Accelerate. So let's say the light Accelerate package, the more intense Accelerate package where you get more support basically, and then the maximum support package where you also have a lot of on-site and telephone support. The best way is to order it via your account manager. That's a short answer.
Okay, Marc, what is the expected resolution performance of the Glacios?
The Glacios is pretty new, so we actually first were not sure what the resolution target was, but we've seen now, what I showed in the presentation was a resolution of 3.2, 3.3 angstrom, but recent experiments that we did in the nanopod has pointed out to a resolution of 2.6 angstroms. So we're really getting to the sweet spot of the resolution. I think for a lot of scientific use cases, 2.6 angstrom is probably also sufficient. Again, if you want to push the resolution scientifically towards the levels in the 2 angstrom range, then probably a 300 keV FEC system like the Titan Krios is needed. But the glaciers or 200 keV FEC in general probably will get you at 2.5 angstrom resolution.
And our next question is, are there any tools or regions on the sample prep side of things that could help improve the workflow?
Yes, so if you talk about the sample prep side of things, then currently what we have to help there is the Vitrobot. It's actually, I would say, a well-established instrument, also proven value. I think we have more than 600 Vitrobots in the meantime in the field. Of course, there will be things that can also be improved on the respect to the Vitrobot. So it's, I think, especially with respect to the connectivity of the sample preparation in general and loading the sample into the microscope, there's still room for improvement. What also actually is an interesting topic of discussion currently in the field is the grid carrier. So that is actually related to the material you put your sample on. Depending on the material, you can get actually a better distribution of your particles, and a lot of research is also currently done into that field. So it's not only the vitrification, but also, again, how can we equally distribute the particles in a vitrified section, and the sample carrier can play a crucial role there as well. And that's currently under research, basically.
XRD requires about 10 milligrams of protein for screening, but how much does Cryo-EM require?
Yeah, I would say a concentration of 10 milligrams per milliliter, that's a good concentration also in general for Cryo-EM. Depends a little bit on the type of material that you look into, but any concentration between 10 milligrams per milliliter is also what is typically used or at least advised for Cryo-EM. So there is actually a similarity in that respect. But luckily you don't need to crystallize the sample, so it's just purified fraction of this concentration is sufficient to start the vitrification process.
And if there are no more questions, Marc, I would like to once again thank Marc for his presentation. Marc, would you like to make any final comments?
No, I hope it was useful to you. As I said, if you want to learn more about Cryo-EM, please visit our website or contact one of our colleagues, and we are happy to help you out and provide you with more information. Thank you for your attention.
I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2019. Please share that message with your colleagues who may have missed today's live event. Also, stay tuned for our next presentation, Introduction to the LabCoat Live Smart Start Training Courses for XB Protein Expression Systems, presented by Stephanie Culbertson. You won't want to miss it. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.